Hello, welcome you all to our channel BioGoogle. Welcome to the new chapter, that is molecular basis of inheritance. Okay, whenever we say the word inheritance, there must be two generations. One, the parent generation, other the daughter generation. So from that parent generation to the daughter generation, maybe from father to son, mother to son, father to daughter, or mother to daughter. So if there is any transfer of the characteristics, like you may say in generally the son is looking like father, or the daughter is looking like a father, or the nose is like his father, the ears are like her mother. So something like we'll just compare between the parents and the offsprings, parents and the children soon after the birth. Okay, that's all because of the phenomenon, nothing but inheritance. Okay, but just the transferring process is known as a heredity, the process where the characters are transferred from parents to offsprings, one generation to next generation, is known as heredity. But the overall process is known as inheritance. Okay, but don't think that all the characteristics from parents are transferred to the offsprings. Only some of the characteristics from father, some of the characteristics from mother will be transferred to the daughter generation. Okay, that's inheritance. The chapter is molecular basis of inheritance. The question is, for the inheritance, what are those molecules which help in transfer of the characteristics, transmission of the characteristics, whether it is DNA, whether it is RNA, or it is a protein, or it is a vitamin, or a tissue, or an organ system, or a molecule. What is that molecule? What is that molecule that is responsible for the transfer of the characteristics from father to son, mother to son, or one generation to other generation? Okay, then for all those questions there is a single answer, that is the nucleic acids. Nucleic acids are the molecules which are responsible for the inheritance of the characteristics. Okay, then what are those nucleic acids? Let us see them properly. Don't make confused, just I have written randomly. Okay, to say that we are not sure about any molecule which is responsible for inheritance. Okay, after some discovery we found that among all of them DNA is the molecule that is responsible for the inheritance of the characteristics from parents to offspring. So don't worry about this schematic representation, right? Then after few studies it was discovered that the nucleic acids are the molecules that are responsible for the inheritance. Okay, we'll see what are those nucleic acids. We have two terms here. We have two terms, nucleic and acid. So why they have given the two terms here? Okay, better we'll understand with the help of a diagram. So we have a cell. We have a cell. Imagine either a plant cell or an animal cell. So okay, cell is covered by the membrane or a cell wall or cell membrane, plasma membrane, whatever it is. Right. Inside the cell, the fluid substance is called cytoplasm. Inside the cytoplasm, there is a major cell organelle, which is known as the nucleus. Inside the nucleus, there is a network of fibers, which is known as chromatin fiber. chromatin fiber or chromatin network. Chromatin network. And this mass of chromatin fibers, a compact mass of chromatin fibers, is known as nucleolus. Okay, nucleolus. So the question is, what is the composition of this chromatin network? Means what it is made of. What it is made of. Okay, here it is made of higher level coilings of the DNA, which are known as solenoids. Which are known as solenoids. The solenoids are in turn are the structures made of a nucleosome, and each nucleosome is made up of one histone octamer, one protein wrapped around by the DNA. Wrapped around by DNA. So here when we observe all these sequential packaging levels, we found the answer that DNA is the major molecule, one of the molecule which has a great contribution for the inheritance of the characteristics. DNA has the greater contribution for the inheritance of characteristics. Okay, you may ask another question: what if we go in reverse order? What if we go in reverse order? Okay, so DNA will undergo coiling around the histone protein to form nucleosome. About 5 to 6 nucleosomes will again start coiling to form a solenoid. Solenoid will again make a higher levels of coiling to make a chromatin. Chromatin will then start to condense to form chromatid, to form the chromatid, to form the chromatid. Lastly, the chromatid will form thick thread-like structures known as chromosomes. Known as the chromosomes. Okay, so hope you all know that the chromosome structure is almost like okay, so this we can say the chromatid. This is the centromere. Okay, and here the end part is known as telomere. And on either side of the chromosome there are 2 disc-like structures. Those disc-like structures are known as kinetochores. Okay, another question arises here: when did the chromatin network form the chromosomes? This process occurs soon before the cell division. Okay, in normal state, in normal cell, the nucleus contains only chromatin network, chromatin fiber. Okay, again there are 2 types of chromatin. One is euchromatin, and the other one is heterochromatin. That's different case, but always it remains in the state of a chromatin. But only during cell division the chromatin undergo another level of condensation. This process is known as condensation. Condensation to form the chromosomes. Right? So here for these chromosomes it is referred as the chemical basis of the physical basis of life, the physical basis of inheritance, physical basis of inheritance. Inheritance. When it comes to DNA, it is the chemical basis of inheritance. Chemical or a molecular basis of inheritance. Molecular basis of inheritance, right? So this is how the DNA is organized into a chromosome during cell division. Again, after cell division, again it comes back to the chromatin state. But don't ask when it comes to DNA. Okay, the chromatin contains DNA in the multiple levels, but it's always remained in the state of chromatin network before the cell division, even after the cell division. But during cell division, the chromatin network will undergo condensation to form the chromosomes, right? That's how the chromosomes and the DNA are organized with each other, either before cell division or during cell division. Okay, and you may ask why this process occurs. Why this process occurs? Because if the cell divides in this state only, there may be unequal distribution of the chromatin network. There may be unequal distribution of the chromatin into the two daughter cells. What are those daughter cells? For example, if one cell divides into two cells, so whatever the material present in this cell, it must be transferred to both of these cells, parent cell, daughter cells. So if the cell divides in this state, there must be unequal distribution of the chromatin, so that the two daughter cells may receive unequal number of characteristics. This may receive more number of genes, this may receive less number of genes, or else this may receive more number of genes, this may receive less number of genes. To avoid that, the cell has developed a mechanism called condensation. Through that condensation, the process of condensation will make chromatin to chromosomes, but the chromosome number will be equal. Usually the even number of chromosomes are formed. Okay, for example, in this cell 46 chromosomes are formed. So when the cell divides, it will get 23 and 23 set of chromosomes, so that the equal amount of the DNA is being divided among the two daughter cells. Equal amount of DNA is distributed among the two daughter cells of this parent cell, right? Then you may ask whether all the organisms have the same type of organization. Like we have a bacteria. Okay, remember, bacteria is a prokaryote. Okay, and we have another organism called amoeba. So recall, amoeba is an eukaryote, but amoeba is a unicellular eukaryote. Okay, though bacteria is prokaryote, single cell, but it is a prokaryote, means with a primitive nucleus. But amoeba, though it is single cell, it is a eukaryote. And lastly, as developed highly organized organisms as human beings, these are the multicellular eukaryotes. So in these human beings, there are different levels of organization. For example, cellular level, next tissue level, organ level, organ system level. Organ system means digestive system, respiratory system, nervous system, excretory system, reproductive system, endocrine system, homeostasis. Like all those organ systems together will make an organism. Together will make an organism. Okay, but when we come to cellular level, this is the organization. Inside the cell we have a nucleus. Inside the nucleus we have a chromatin. Inside the chromatin there are solenoids. Inside the solenoids there are nucleosomes. Inside the nucleosome there is a large number of DNAs or large amount of DNA or very length of DNA. Okay, so this is the basis of inheritance. Means these are the molecules of inheritance. Okay, you may ask another question. Why it is given the name acid? Yes, it is present in nucleus. You have got given the name nucleic. Why is it given the name acid? Okay, when we say the word nucleic acid, there are most commonly available nucleic acids as DNA and RNA. Okay, they are present in nucleus. Yes, so nucleic. Why is it acid? Why is it acid? Yes, your question is right. Just because DNA and RNA both are made of three components. Both are made of three components. First component is pentose sugar. Pentose sugar, that is ribose. Second component is nitrogen base. Nitrogen base. Third component is the phosphate group. Phosphate group, that is phosphoric acid. Phosphoric acid, that is H3PO4. So phosphoric acid. Okay, so this acid makes the word nucleic acid. Okay, the genetic material DNA or RNA belong to nucleus, nucleic. They have an acidic component, acids. So the name nucleic acids, right? So this is actual base of this chapter. Okay, so in this chapter we are going to study all about the DNA. Okay, there are some experiments which prove that DNA is the genetic material. Okay, because some people can say that RNA can also be the genetic material. Yes, RNA is genetic material in some viruses, not in all the organisms. RNA is genetic material, but in not all the organisms. But RNA is genetic material in only few of the viruses. They are called RNA viruses. And most of the viruses have a DNA. They are known as DNA viruses. So virus having RNA is known as RNA virus. Virus having DNA is known as DNA virus. Okay, except those viruses, remaining all the organisms have a DNA as the genetic material. Most of the organism or all of the organism except the RNA viruses have a RNA as the genetic material. Okay, so in this chapter we will discuss about the chemical composition of the DNA. I mean detail about all this. Then structure of the DNA. I mean how these chemicals are arranged among themselves. Or there is a most famous model called Watson and Crick model. We will discuss about that in this chapter. And most importantly we discuss about the packaging. This process is called packaging. Means from the DNA how the chromosomes are formed. How the chromosomes are formed, whether by coiling whether by supercoiling, whether by depositing on itself or any other compaction process. So, this sequence is known as a packaging of the DNA, means how DNA will form the chromosomes. Okay. So, we will discuss that also in our in further videos. Okay. Then we will also discuss about the replication of the DNA. Replication means if there is one copy of the DNA, it has to form two copies. Means the amount of DNA is doubled. The process is called replication of the DNA. Okay. Then we will also discuss about the transcription process, conversion of DNA into RNA, then translation process, RNA to, better we will write it on the board. So, if you want notes, better pause the video and take it down on your book, otherwise it will take time for that. So, better pause the video and take the things into your book. Fine. We will just see that what is molecular dogma, and we wind up the video by studying the central dogma of the molecular biology. Okay. Means we said that it is a transcription when DNA converts into RNA. It is called translation when RNA converts into protein. Okay. So, it is called by the famous name, that is a central dogma of molecular biology. Central dogma of molecular biology. Okay. Or it is commonly known as gene expression. Commonly known as gene expression. Let us see how this expression of the gene occurs. So, the DNA present in the nucleus converts into the messenger RNA. Then the messenger RNA converts into the protein. Okay. Here the process where DNA converts into mRNA, it is known as transcription. And the process where the mRNA converts into protein or will form the protein is known as translation. Okay. Or the process where DNA forms by using DNA, means the process of replication, a process by which one set of DNA will form another set of DNA is known as a replication. Okay. When DNA forms RNA, it is called transcription. When DNA forms DNA, it is called replication. Okay. So, here replication is also known as autocatalysis. Transcription is also known as heterocatalysis. Hetero means different. From DNA, another molecule is formed, that is RNA is formed. So, it is a heterocatalysis. But in replication, DNA is formed by DNA. So, it is a autocatalysis. Right? And this process have a various location in various organisms. Okay. When it comes to eukaryote, plants, animals, higher animals, the transcription takes place in the nucleus. Transcription takes place in a nucleus and translation takes place in a cytoplasm. You may ask another question. In case of eukaryote, transcription in nucleus, translation in a cytoplasm. What if the organism is a prokaryote? What if the organism is a prokaryote? Okay, we'll draw two different cells to differentiate among the two processes and their location in a prokaryotic cell as well as a eukaryotic cell. Okay, let us take a bacteria. Take a bacteria. It's a prokaryotic cell. Cell, that is bacteria. Okay, and we'll draw one plant cell or animal cell. Plant cell or animal. Imagine it's an animal cell. So it is a eukaryotic cell. Okay. So in eukaryotic cell, as we discussed at the beginning of the video, there is a well-defined nucleus. Well-defined nucleus. But in prokaryote, there is no nucleus. There is no organized nucleus. Means here the genetic material is covered by nuclear membrane. But in a prokaryote, there is no nuclear membrane at all. Okay, it has DNA. It has the DNA, but it is not covered by any membrane. But here the DNA is covered by the nuclear membrane. Okay, so this is DNA. This is DNA. Okay, so in prokaryote, DNA converts into mRNA. DNA converts into mRNA. The process known as transcription inside the cytoplasm itself. But here the DNA converts into mRNA. Inside the nucleus. Coming to prokaryotic cell translation, the mRNA will help in the synthesis of a protein. So proteins are synthesized inside the cytoplasm itself by the process called translation. DNA to mRNA, transcription, mRNA to protein, translation, everything inside the cytoplasm in prokaryotic or bacterial cell. But when it comes to eukaryote, the DNA is inside the nucleus. So DNA becomes mRNA by the process known as transcription. But you should also remember that for the process of translation, there is a need of one of the important cell organelle called ribosome. These ribosomes are present only in the cytoplasm. Okay, so in cytoplasm of this, there are ribosomes. They will help in the translation process. Here, ribosomes are present in the cytoplasm. mRNA is present inside the nucleus. So now mRNA has to come out. Of the nucleus to the nuclear pore. Then only the process of translation starts. So now the process of translation, that is conversion of mRNA to protein, will take place. This is the protein. This is again mRNA. Just because prokaryotes have a ribosomes in cytoplasm, so transcription occurs in cytoplasm, so there is no need of any transfer of a mRNA. But in a eukaryotic cell, inside the nucleus, the DNA converts into mRNA, but the nucleus does not have the ribosomes. Because of that, the mRNA has to come out of the nucleus to the cytoplasm, where the mRNA binds with the ribosomes, so that it can undergo translation to form the proteins. This is the difference between the prokaryotic cell as well as eukaryotic cell in terms of the location of the transcription and the translation, right? So this is how the gene expression works. Okay, this is very important phenomenon in all the living organisms. So remember properly, study properly. That is known as central dogma of molecular biology, or also known as a gene expression, where DNA by the process called transcription becomes mRNA, and then mRNA by the process called translation becomes a protein. Okay, in prokaryote it is both processes take place in cytoplasm itself, just because there is no compartmentalization. There is a best word called compartmentalization. Compartmentalization means there are no compartments like cell organelles. Here there is a well defined compartmentalization. Compartments means organelles, nucleus, ribosomes, endoplasmic reticulum, Golgi body, mitochondria, chloroplast, etc. will have means they are the compartments or the definite organelles or the definite chambers in the cell. Okay, that is how the processes work in a prokaryote as well as a eukaryote, right? So in the next video we will discuss about these three components, and then we will move on to the structure of DNA, or we will discuss in the further classes about the packaging of the DNA, etc. as per the schedule or as per the concept, right? So this is enough for this today's video, and one request is, if you are new to my channel, please subscribe to my channel, and if you find this video really useful, please share the video among your friends, students, or anyone who are there in this field, who are willing to learn the things. Okay, there is no loss if you just press on that red colored subscribe button. Okay, if you have any more doubts regarding this class, just add a comment, add your question in the comment box, so that I will be able to reach you back by your answers. Okay, till that, keep learning. See you in the next video. Bye bye.